Welcome to Tech and Drugs, the podcast where we explore how data and AI are transforming pharma and biotech. I'm your host, Thibaut Giroux, scientist, technologist, and lifelong connector of science and data. In each episode, I sit down with innovators pushing the boundaries of drug R&D to uncover the real impact of AI beyond the buzzwords. Today, we are excited to welcome a fascinating innovator in AI-driven drug discovery, someone who has been at the intersection of computational biology and pharma innovation for decades. Douglas Selinger is the founder and CEO of Plex Research, a company pioneering a novel AI-driven analytical platform designed specifically for scientists. What sets Plex apart is its commitment to transparency. Scientists have direct access to the underlying data, ensuring that AI-driven insights are both explainable and actionable. Doug is no stranger to large-scale data analysis. As an early pioneer of microarray technology, he offered some of the first publications on experimental and computational approaches for large-scale transcriptional analysis. After earning his PhD in George Church's lab at Harvard, he spent 14 years at the Novartis Institute for Biomedical Research, NIBR, working across the entire drug discovery pipeline, from target identification and validation to high-throughput screening and preclinical safety. In 2017, Doug founded Plex Research to develop a new form of AI inspired by search engines algorithms. Plex platform integrates vast and disparate datasets, small molecule bioactivities, gene expression profiles, genomic, proteomics, metabolomics, etc., to reveal hidden biological connections. By making sense of massive chemical biology and omics data, Plex helped biotech and pharma companies accelerate drug discovery in areas such as target ID and validation, disease identification, biomarker discovery, safety assessment, precision oncology. So how is AI truly shaping the future of drug discovery? And how do we move past the hype to deliver real, interpretable, and actionable insight? Let's dive in. Doug, welcome to the show.
Thanks, Thibaut. Thanks for the invitation. It's really great to come here and have a chance to talk to you about this topic.
We'll talk about the company, we'll talk about AI, data, of course, but I like to start with getting a little bit more history and insight on the person I'm talking to and understand how did you get to where you are today? What brought you to this field? Can you tell us a little bit about the early days?
Sure, yeah. So I'm a molecular biologist by training. So as you mentioned, I did my PhD at Harvard in George Church's lab, which is a really fun place. There's a lot of cool things that come out of there. It's really exploratory, all kinds of crazy stuff. And I was there at a really interesting time when genomics was coming out. So we knew genomes and the catalog of the parts list, all the pieces of the cell, all the genes. But the question was, how does it all work together? How does it function? What are the dynamics? Can we do that on a large scale? So I was working with some of the early technology, microarray technology, so looking at gene expression on a large scale. And I was in the lab actually getting that to work. And once we had the data, now we needed computation, we needed software, and there was no software at the time to analyze that kind of data. So I shifted to computational approaches. When I graduated, I went to Novartis when they first moved up into Cambridge, Massachusetts, and worked on all kinds of large-scale projects, both as a computational biologist, also running a large-scale transcriptional profiling platform. So both computation and experiment over many kinds of drug discovery projects. So I really got a flavor for all the kinds of approaches that are being used in drug discovery and the interface of large-scale biology and computation in the drug discovery process. At some point, I hit on this idea that we have all this data. So at a big company like Novartis, certainly there's a lot of data. And in the public domain also, there's a tremendous amount of data just in the public space, in these databases and in all these supplemental files that accompany all the scientific literature. And I hit on this idea that Google had tremendous success with internet search. So taking the massive amounts of information on the internet and looking at using the structure of the internet, what are people linking to, for example, and using the things that people link to decide what's relevant, what's most important, follow the links and see where they converge. And what I realized is that you could take that kind of approach and apply it, instead of web pages, apply it to data and ask across all the world's data, where does it point to a specific answer to a scientific question? And so is there more in the data than we're currently seeing? So does the data actually point us to answers that we weren't aware of that might not even be in the text of papers? So long way to say, there's this really interesting idea, this new approach. Ended up leaving Novartis in 2017 to found Plex. And here we've been building a system to really take that to use that on a tremendous scale. And we've been working with lots of companies at this point, over 40 companies, biotech and pharma, helping with all kinds of scientific problems using this very general approach. That's, if we look at the world of data, all the world's data in principle could be put into this sort of system. We can ask, here's my question, what does all that data have to say about it, right? So what's the best answer, or the answer with the most support? And then what's the actual data that supports that answer or that discovery?
So we'll come back to Plex in a moment. But one thing I've seen sometime with entrepreneurs is that they start something out of frustration in a role that they had. So I used to work with someone who was managing data at a large pharma, and the data was a mess. And he was like, okay, we need tools to fix that. And if we have this problem here, most probably everyone has the same problem. So was there any specific frustration that drove you to try to fix this frustration?
Oh, that is a great question. Yes, tremendous amounts of frustration. So there's a couple categories. So one is, so I've seen so many panels, and I've been on some of these panels too, but mostly seen these panels where people talk about data silos, that we have all this data and we want to somehow use it to do computation and to make it machine readable and usable on scale and all of these things. And the panel comes up with, if only the scientists, because they put the onus on the scientists, if only the scientists would generate more data, cleaner data, better data, the right type of data, the better annotated data. It's always the same. It's been the same list of complaints forever. And it's not to say we haven't made any progress. We've made some progress, but it's still the same issues. And what the point that I raise is that maybe it's the algorithms. Maybe we need some algorithm that can handle the data as it is, right? The data we have now. I've never gotten a call from Google saying, your website, could you take down your website? It's really screwing up our algorithm, right? No, it's robust to noise and to whatever people put on the internet. It does not impact, or has very little impact on the results. So you need an algorithm that works with, or an approach just generally, that works with the data that we have. You can't perennially wish that we had different kind of data. And if you let me, the other big source of frustration is that we have this disconnect, or we've had for a while now, a disconnect between our publication process and our technology. So for a long time now, we generate huge amounts of data. And I think scientists know this. I'm not sure how aware the rest of the world is, but certainly scientists in this field realize this is happening. Perhaps we've compartmentalized it and stopped paying attention to it. But we write papers, we give presentations, and there'll be a slide, let's say, with thousands of data points, a scatter plot, a heat map. We have these dimension reduction plots, UMAP, Tisney, all these crazy things that have thousands, and data points are sometimes behind them. Each data point represents 1,000 data points. So we routinely just show these massive data sets, but the amount of information we extract from that data is tiny compared to the amount of data that's in the figure. So there'll be a figure with 1,000 or 1,000,000 data points, and then we'll summarize it with a sentence or two, right? So somewhere we know that we're missing out, that we are not capturing even the tiniest fraction of what we've collected, the data we've collected, the tiniest fraction that is actually turning into knowledge. And we know that, but I don't think as a field, we know what to do with that. And that was another big source of frustration, that I would sit through these, and I would see this kind of presentation and realize that, wow, the data set that was generated right there contains so much information that we're not leveraging. This is a huge opportunity that's just being wasted because we just don't know what to do with it. And both of those problems that I laid out are things ultimately that Plex is addressing. And I think we really have an approach that fits both of those problems. And so it was truly born out of frustration, certainly.
But I worked on a few projects where we were looking into a historical data set to train models. So we had collaborations where we had companies that had very interesting predictive models, but of course, to improve those models, you need to train them with a lot of data. So we were going into our archives looking for the data, but we faced a number of problems. First of all, to locate the data. And then when we have the data, to actually make the data AI ready, for the lack of a better word. Very often we will start to look into the data, and that will be horrible. You open the file and you have just a bunch of numbers, you have no annotation, you don't know what it is. and became maybe a bit cynical about the value of historical data, because my impression was if you go back too many years, then you start to have something which is so dirty and so unusable that sometimes it's cheaper to reproduce the data. I don't know if you ever experienced something like that.
Certainly, there's a lot of data that perhaps is too noisy and too maybe should be reproduced. But there's so much data that is high quality, that is even that's in the right format, that's the right kind of data, that checks all the boxes that I mentioned before. There's still an incredible amount of that. So even if we set aside, yes, of course, there's going to be cases where it's difficult to get the data and there's all these reasons, all these barriers, there's still a tremendous amount of potentially low-hanging fruit, a tremendous amount of data that is there and ready and high quality and usable and all of that. So there isn't really a lack of data. And even of that data that is usable and checks all the right boxes, we're using very little of that. And by very little, vanishingly small amounts of it, right? Because again, you have your millions of data points and you summarize it in a sentence or two, these sorts of things. So we're making, even with that issue, we're still making use of the tiniest fraction of what we have, right? And then also when, let's say you do have a case where you've assembled all this data, you've trained your model and it works well, right? And now you have a useful model. Where does that model go? How is that being accessed, utilized? That's a big issue too. That's a big problem. Often I'm going to email the person to build the model, ask them to run it for me and send me the results, email me the results. This is not going to scale. And there are better solutions than that, clearly, but there's not a way right now to just say, I want all the world's models to be run on this, and wherever they're relevant, give me the answer. Most of these methods are just lost because there's not a way to uniformly access them, use them. And even when you do use them, often they're only useful for certain niche cases, certain cases. And you do want them to come up when they're useful, but you don't want them to come up in other places. So for example, there's a lot of pharma companies will train models on activity. They'll say, here's a bunch of chemicals that we have in our archive, and we've tested them against a protein, and here are the ones that are active, so they actively inhibit the protein. Right? And we take all that data, and now we can predict for a new molecule whether it's going to be active or not. And that can be useful. However, the utility is limited to that specific question, right? So is it going to be active or is it going to inhibit? So this idea of activity, if you have someone who says, I want to know, I don't even care if it is going to inhibit, change the activity, I just want to know if it's going to stick to it, if it's going to bind to it. Now, this is a whole different question, right? Not an entirely different question, but different enough that the model you built that predicts activity may or may not predict binding, right? Because that's a different thing. So I just use that as an illustration that the models they're only predicting what it is you train them on. If you train them on one thing, they won't generalize. You have to at least pay very close attention to how you're asking them to generalize, right? So there's a lot of these nuances that are important, and they can get lost, and they could limit the utility of machine learning in certain instances.
I want to go back to something that you said earlier. When technologists, data scientists, and data scientists work with scientists, sometimes they complain and say, oh, why haven't the scientists labeled the data or prepared the data in a certain way? And my experience is that very often the scientific process in pharma organization is very linear. You have a scientific question, you prepare your experiment, you run the experiment, generate the data, analyze the data, and then you move on. And then you keep the data. If you're lucky, you keep the data because of regulatory stuff. If you're not lucky, then you just lose the data and move on. And of course, now things are changing because we start to think about data as a reusable asset. Or maybe we think about that. I know a lot of scientists who don't think about data as a reusable asset because they still think about the traditional way of doing research and not the way that this lab in the loop concept that starts in a model, the model informs the experiment, then the experiment generates data that feeds back into the model. And of course, if you want to do that, you need to have the data prepared in a certain way. Going back to something very fundamental, what do you think in pharmaceutical organization is the biggest obstacle to get those different people who have different background, different language to actually work together, the technologists, the data scientists, and the actual life scientists?
I think the technology needs to keep up, needs to solve this problem. I think this is really a problem that ideally could be solved by an algorithm, right? Because if you think about web search, before Google came around, basically invented PageRank, which is the algorithm that made Google. We're going to use the structure of the internet to figure out what's relevant. Before that, there were all kinds of things on the internet that you couldn't find, that no one knew about. And so the motivation to put things there or to look there was really reduced compared to now, when you can find everything. Now people are going to put things there and people are going to look there, and you're just going to get more value. So I think also with the scientific enterprise, if we can demonstrate, here's an algorithm and an approach that when you generate some data, its utility will go on and on. It will be reusable. It will be caught. You don't have to jump through extra special hoops to annotate it and generate it in certain ways. Otherwise, it'll be lost. Something where the algorithm can do most of the heavy lifting, so that the scientist does what they do. They generate the data as they do it. The algorithm or the approach takes care of the reusability. And that's what I think we've hit on here, that it is so different and so impactful, is an algorithm. And again, we didn't really invent it. So Google showed this works for internet search, and many others have shown that this kind of approach works for internet search. All we're doing is repurposing and saying, it also works for data analysis. You can point this at data, and you can now suddenly reuse incredible amounts of data that have been produced. So that's really so I think the onus is on the algorithms, because clearly we've spent literally decades yelling at scientists or berating the scientists for, again, the list of not enough data, good enough data, not annotated enough, not the right type of data, on and on. There's only so much you could do to change the scientific enterprise itself. This is complex stuff that scientists are measuring and capturing. There's a reason why it's been so hard to make it uniform, especially in this space, in biology and drug discovery. So that's why we're so excited that we think there is an answer, right? From another field, right? Internet search. There's an algorithmic approach that really addresses this, that can unleash just an unfathomable amount of data that we've already collected, that's sitting there, that could be reused if only we could figure out how to reuse it. And now we have a method that can reuse it.
So it's a good segue to the next part, because we talked about the relation between technologists, data scientists with the scientists, but I think we can look at it from the other way, like scientists, how scientists think about technology, think about AI. And obviously it's everywhere at the moment. When was it? I think earlier this week, there was NVIDIA GTC, the big annual show from NVIDIA, where a significant part was on life sciences application. There was healthcare, but also hardcore drug R&D, after the robot and the chips and all those things. And I could see that, because it's interesting to see the focus and the investment of really large tech company, $1 trillion company, in drug R&D, in making an impact in drug R&D. That being said, there is a lot of skepticism when you talk to scientists. So you will have some of the scientists that are obviously interested, they are fans of AI, and then you have the exact opposite, like really skeptical. I don't really see a lot that are in the middle, see the good, the bad, and are a little bit more moderate about AI. I think it's one extreme or the other. So talking about your company, Plex, because you play in this area, so how do you ensure that the AI-driven insights are both reliable and scientifically rigorous? Because that was my experience working within pharmaceutical company when I was introducing tools to some of my colleagues. You just have one chance. If when you do a demo, things is not really accurate, you don't get the thing that they expect, then very often people will say, oh, you know what, come back in a couple of years when your thing actually works.
Yeah, there's a lot of skepticism, and I think it's justified, to be honest. I'm skeptical, knowing people who understand machine learning, right, know the limitations and know what kinds of data you need for those approaches to work, and the conditions and the context in which they're successful. And so if you then go into a team of biologists or chemists and you tell them, okay, this is going to solve all your problems, it really depends on what the problem is. And it really, to fit into machine learning, it has to have certain very specific properties. So if you walk in the door with the example I talked about before, you've got a model and it predicts activity inhibition of your molecule, right? And you go in and say, okay, I'm going to sell you this tool now. It's going to make predictions for you about activity. You have to be really clear that maybe they want to know binding. Actually, the scientist says, I want to know binding. Activity is related to binding. It's not quite the same thing, right? So there's all this hedging. And then the question is, what was your training set? Because maybe we have brand new chemistry, nothing like it in your training set. Now the model's not going to work either. So now your model works in a very narrow domain that doesn't capture what the biologist is trying to do, right? So either the technologist has to go in there aware of that and not oversell, in which case no one gets all that excited about it, or they oversell and overstep the actual utility and they get called out and they get caught because the chemist or the biologist, like, no, I wanted binding. No, my chemistry is too different for you. And you should have told me that wasn't going to work. And there's all these, I think that's what's leading to the dynamic, the skepticism. I think it's real. What we've done, and this is the reason for our approach. So I've been frustrated and scientists have been frustrated by that dynamic. So we're doing something entirely different. So we are not making predictions, for example. Strictly speaking, our platform is just finding things. It's finding data. And it's just returning for your question. Here is where there's data, the most data. Here are the kinds of things that data most points to, most converges on. And here is that data for you to interpret. So if you're interested in binding and you look at the data and it's binding data or it's not binding data, right? Some other kind of data, what to do with that, right? So the details are important. The transparency is absolutely critical. And that's what we're providing. So we are allowing scientists, basically giving scientists superpowers, giving them a capability to find things that they otherwise couldn't find. And once they find them, because we're providing all the details, they can evaluate it. They know what to do with it, right? So it's a very different dynamic that's much healthier, that's much more aligned with the scientific enterprise and what scientists need. And so I think once they get a feel for this approach, a lot of the skepticism is going to melt away, right? Because you're going to see, oh, this isn't someone coming to us and saying, here's a prediction and look at the p-value, the significance, the confidence, whatever measure it is, which we don't really know what to do with. Instead, it will be, here's something we discovered, here's something we found, and here's the data, the experimental data that we found that supports this conclusion. They know what to do with that. And I think when they start seeing that and seeing that, oh, that data came from all these different places that maybe they were theoretically, they could have been in the seminar or read the paper that generated that data point, but there's no way they're going to remember it. We're assembling it all and saying, here it is. Again, data that may not have been in the text, even not even mentioned in any of the summaries or any of the text. It was just buried in the actual raw data. We're surfacing all that, saying, here's where the data points actually, and here's the data. So it's a healthier dynamic, and that's a better fit for the scientific enterprise.
Is there any real world example that you are at liberty to share with us where you worked with customers and help them uncover some critical insight or things that we will clearly not have found without the help of your solutions?
Yeah, absolutely. So we've been around for almost 8 years now. We've worked with all kinds of companies, more than 40 companies, biotech and pharma companies. Most of what we do with the companies we can't really share, unfortunately. But this last year, we've started working more with academia, and those are more things that we can talk about. And we do have a preprint that we just released that has a number of examples in there as well, explicit examples. But for one example that we've been talking about, so we collaborate with a group in University of Michigan, Johnny Sexton's lab at University of Michigan, and they run phenotypic screens, which just means that they set up experiments and they test lots and lots of compounds and chemicals. And they ask, can I find some that have the effect on my biology that I care about? So I'm going to try each one out and see if it does the right thing to my cells that indicates a potential therapy, right? So now you've got a bunch of compounds or chemicals that have interesting biology. Maybe someday they could be a therapeutic because they seem to do the right thing to the biology. But the problem is that we don't know how they work. We just know you put them on the cells and it looks great, but no idea how they work. And it's really important to know how they work. So we were working with them and taking their compounds that are hits in these kinds of screens. So one of them focused on liver biology. So one of these compounds seems to be promising as a way to treat fatty liver. Works in mice, but again, they don't know how it works. So they came to us, we put it in our system, and we said, these compounds, the chemical structure of these compounds is similar to these other compounds here that we found out in the world in all these databases. And those compounds are known to inhibit certain targets. In fact, DPP4 was one of them, and one of these HSD targets, these hydroxysterol desaturase targets. And they tested both of them. Actually, the first one, DPP4, confirmed inhibition of that target. So one of the mechanisms that we proposed, they confirmed, and they presented that data in a conference in October in London, the LRA conference. And recently we got word from them that they confirmed the other one as well, the other mechanism that we proposed as well. And that's work that we're working on publishing with them. So that will be coming out hopefully in the near future. So we've done a lot of work around mechanism. Ultimately, that's, I think, where we've had the most impact is we can use the world's data to try to understand mechanism. So mechanism of disease. How does this disease work? What are potential therapeutic approaches that we could use to address this disease? Therapeutic mechanism. Here's a drug that works, or we think it will work, and we have some evidence that it treats a disease or helps with the disease. How does it work? So our system can help identify mechanisms of therapeutic response. Also, then that leads also to who should we give it to? Because maybe it will work for some of the patients, but not the others. We do a lot of work in precision medicine where we identify based on understanding how it works exactly, understanding which patients will respond, which ones won't. And then finally, safety. First of all, if we see a safety issue, can we understand it so we can fix it or avoid it or not have it in the future? So safety assessment, mechanism of toxicity, mechanism of adverse events. So we address that as well. And ultimately, I think all this leads to more effective precision medicine, more effective, basically discovery of cures, right? Because when we discover a new therapeutic mechanism, that's what we're doing. And also more safer medicines, right? So fewer side effects, not giving it to the patient who's going to have an adverse reaction.
So when we had a chat last time, you shared with me a paper. So is it this paper that you're talking about, the framework for autonomous AI-driven drug discovery?
That's right. That's our preprint. Yes. Yeah.
Yeah. So we will share the link together with a podcast. So you talked about a number of things like the complexity of the data and the complexity of linking the different concepts. So maybe can you walk us through, you talked a little bit about it, but can you walk us through some of the key challenges when people are faced with data overload like that?
Yeah, so I think reading these papers and attending scientific conferences and just seeing this data flying across the screen and knowing we barely touched any of it, it's very frustrating. I'm not the only one. I know that I'm not the only one that's frustrated by it. So what's really exciting about this approach is that it checks a lot of the boxes as far as a solution. So if we were to solve this problem, we would want an approach that works on really diverse data. This field has incredibly diverse data, and doesn't seem to be changing anytime soon. Diverse data, very complex data, noisy data. So it has to be very robust to noise. So it really has to capture all that, and we want it to ultimately give us a concise answer, because we can't, if we just put in massive amounts of data and then our result is a long gene list or compound list or whatever kind of list, that's not helpful. We need something that says, no, this is the top thing to look at, maybe this is the second thing. So it's got to be really concise, and then ultimately whatever analysis we do needs to be transparent, especially in science and for regulatory purposes too. We need to know, here's the reason and the data that led us to this conclusion. So I think those are many of the boxes that we would want to check in a solution to our data overload. And again, that's why we're so excited about this approach. It checks every one of those boxes, right? And the concision, because there's always a top hit, and the transparency, we see the data behind it. There's robust to noise because it's consensus-based, so the noisy data doesn't agree. We just look at where there's agreement. So it checks a lot of those boxes for data overload. And maybe we'll get to this. I'll let you respond. I hope we do get to where this all leads, but as far as autonomous research, but maybe we'll get that in a little bit.
Yeah, we'll get to that. So from what I know, Plex has developed a focal graph to tackle the issue of data overload. How do these next-gen knowledge graphs help filter and refine the drug target hypothesis?
That's really the insight that we had, was this idea of a focal graph. So basically what it is, there's knowledge graphs, and a lot of people have worked on knowledge graphs, and you've spoken a lot about it.
Yeah.
So this idea that you can represent knowledge in a graph, so things connected to other things. is in networks. It's a very flexible, robust, comprehensive sort of data model, sort of essentially a way to capture all kinds of things in an organized way. So lots of people worked on that. And as I mentioned, Google and internet search has been able to take large graphs, right? The internet is a graph, is a network, and figured out how to find what parts are relevant. And so what we did is we figured out a way to connect them. And that's what the focal graph is. So what a focal graph is, we take this big knowledge graph, which could be just absolutely massive and incredibly diverse. And what we do is we pluck out from that a subgraph, a piece of it that's relevant to your question, to your query, right? So now you go from an absolutely massive graph to a far smaller, probably still big, but way smaller subgraph, this focal graph. And you've plucked out the bits in such a way that you've captured the bits that are most relevant to your question, right? So if you care about chemistry, then you pluck out the bits that have compounds with a similar structure or things that are related to your query. So now you have, that's what the focal graph is. It's meant to, let's pull out the data that is focused on your question, and then further use the centrality algorithms, things like PageRank, these internet search algorithms, apply them to the focal graph such that now the top ranked member, the thing that the focal graph most agrees on, is a putative answer to your question. And that's the concept there.
Sometimes when you're searching for something, there is a number of data sets that will point you in one direction, and then there is this incredibly rare data set that points you in a different direction. And that's actually really interesting. And it might come from a new publication, so something that is very recent. And if you look at some of the modern tools like the LLMs are really good at, when something is frequent, it will come up because it's a statistical approach. But if there is something which is very rare, most probably you won't find it because it's not how they are built. With your approach, and I don't know enough about how PageRanks work, but that was my impression, that to find something, you need to have many things pointing at it. So imagine if you have something that is very new, that is very rare, but that is very important. Will people be able to find it? I don't know if my question makes sense, but the best way I can formulate it.
No, it makes a lot of sense. I know what you're getting at, and it's a great question. And maybe it's that if there's something unknown, something rare, understudied that we want to understand, and if you have a method that looks for lots of different places all agreeing on an answer, how in the world are you going to find something new? Because you're looking for what everyone knows, right? So that seems like a paradox. What makes it the way that we find new things is because, keep in mind, our approach is actually mostly focused on relatively raw data, right? So the places people have described the information is usually it's in a text or patent, something like that, where it's been digested and someone's made a discovery and they described it, right? So that data is out there, and we certainly have that, but that's a tiny fraction of the data that we're capturing. The vast majority of the data we're capturing are these relatively raw data points, right? So I treated patients or cells or something, and I measured all the genes, tens of thousands of genes, and these went up and these went down, and I changed the dose, and these went up and these went down. So really relatively raw data of all different kinds. So it's actually usually, or very often at least, not known what all that data even points to. And it's certainly possible, and we see this often, and which is a good thing, that the known answer does often come up at the very top or near the top. So we can rediscover from the underlying raw data some things that are known, but we can also discover things that are entirely unknown, right? And we're discovering because it's consensus in this raw data. So it's not that everyone already knew it, and so we're showing that we're saying that the data knew it, in a sense. This raw data, it was there. And in fact, it's the strongest signal. Lots of data people collected all over the place, all agrees that this is the answer. But there may be not a single paper or patent or any written description that actually captures it. So the data knew it, but we didn't know it. And that's the way that we're discovering this kind of novelty. And things that we think of, you mentioned like rare disease and things that we think we don't know much about, given the amount of data that we're not using and that we can now use with this sort of approach, a lot of things that we think are rare, we think we don't know anything about it, are going to turn out, and we're starting to see this, it's going to turn out that we know a lot about it. We know way more than we thought, because in fact, it's been measured many times. It's still the same. People are measuring the same tens of thousands of genes every time, and they're focusing on little bits of it. We're going to start seeing that the answers to things that we think we don't know anything about, in fact, we know a lot about them. And here's the answer, and here's the data that speaks to it, right? That no one knew about. So it's really going to change how we think about how much we know. There's a lot of cures for diseases that are just sitting in those data sets that we can now expose and say, here is the answer. Here's how we know the answer. And obviously, there's always more to do. So people ask me often about validation. Certainly, there's always more experiments to do. But the kind of things that we're producing, again, are not predictions. They are, here's the data we found, experimental data, the vast majority of it, experimental data that points to this finding. Here's the finding, here's the data. That data is often the same kind of validation data that you might then go off to reproduce. So it's along the continuum of validation. So we're going from sometimes zero, where we think we don't know anything, and we're running our system and finding, actually, we know a lot. Here's what we know, here's that data. And if you wanted to do a new experiment, here would be the next thing to do, because again, there's always more to do. But we can often go from zero to, or we could potentially go from zero to a lot. In the paper, in the preprint, we show how to go from zero or next to zero to something, right? And we run it and show you, here's how we can take something where we think we don't know and take the first step in a transparent way. And clearly we can then run that more and build up cases where potentially we have very compelling experimental validation for a brand new discovery.
Sometimes we think that we don't know, but we know. It's just that we haven't looked at it properly. So I was recording an episode a couple of months ago with Ben Selinger, who used to work at Roche. So he was a senior leader in one of the data department at Roche. And Roche now is quite well known for having invested a lot in their data and digital transformation. So he was telling us the story of how it started and how they started to get interest from the senior executive who will end up paying for this digital transformation. And he was saying they partnered with scientists, with a therapeutic area leader who had scientific questions that they said, okay, can you look into our old data and find interesting signals that we overlooked in the past? And that's what they did. They went back, did a lot of data cleanup, got the data, and then when they presented, they explained the methodology. It was during a long day of presentations. So it was telling me most of the executives were on their phone, were not really interested, until they said, we cleaned up the data, looked into the data, and identified a signal, a biomarker that we didn't know we had. And then he told me, okay, everyone put their phone down and said, what did you find? And then he told me that from that point on, they never had to ask money again, because people were convinced that there was value in going back to the old data and looking at it from a different perspective.
Certainly, yeah. I think that's how you get scientists' attention, is you show them data, right? I think there's a lot of understandable skepticism about making predictions, about models and predictions, right? So if you instead come in and say, so here's our conclusion, basically lay out something very similar to what they might see an experimentalist might present their latest findings, right? That they give the background and say, okay, when we ran this experiment, here's the result we get, and here's our conclusion based on all the data that we've collected, right? It's just that in our case, we're collecting data that's already been generated. Yeah. So it's not to say we don't need to go back to the bench and keep generating more data, but the data that we've already generated is just filled with all kinds of discoveries waiting to happen, all kinds of results, that many of which could be quite compelling. And it's just, they haven't been uncovered before, right? So once we uncover them, there's no convincing specific that needs to be done. It's just the standard scientific exercise of, here's data, what do we think this data means? Does it support the conclusions? And what do we do? And where does this bring us? So it's something that scientists are much more comfortable and used to doing, and they see the value in it immediately.
Is there a type of data set that is better than other at uncovering hidden connection, hidden patterns?
That's a great question. I'm sure there is. Ultimately, I think science, we want to see convergence, ideally, of very different approaches, right? You'd like to see very different approaches yielding the same answer. So you ultimately would really like a diversity of data that all agree ultimately on the answer. So I think some data types are going to be more valuable than others, have better information content. A system like this, actually, you can, I don't know if it's run it in reverse, but you can certainly do a retrospective analysis. You can say, okay, we run the system and we found the following really exciting finding or many findings. And we can say which data sets contributed the most to those findings, right? On the one hand. And on the other hand, you can also say what data sets, if we were to add them, would be most complementary to what we already have, right? And there are certainly systematic ways to ask both of those questions of the system. And by doing that, we can say, let's generate more of the data set that was most valuable. And let's also generate data that fills in our gaps. And we can use the system to define both of those very, very systematically. Because not only as far as efficacy, we could throw in a cost piece as well. We could say, what data type has the best bang for the buck, right? Most cost effective. That not only is information rich, it's also cheap, right? What's the best way there? And then that enables the system to get even better, right, at a rapid pace.
Okay, so let's look forward, look in the future. So what are some of the key trends that you foresee in the industry in the next couple of years, and obviously particularly at the intersection of tech, data, and life sciences applications?
We've all seen large language models, right? Just burst on the scene, and just it's incredible what they can do. I think it took most people by surprise, the capability. And so there's tremendous excitement there. And what we're excited about is that technology is incredibly synergistic with what we're doing, right? So if you're looking at large language models, there's a lot of excitement about agents, for example, or retrieval augmented generation, these kind of RAG approaches where you have the model, but then it goes out and it asks questions, it runs searches, it uses tools, right? These agents. So we've seen that these models can be tremendously augmented. For example, if they run an internet search and they find what's on the internet and incorporate that. What Plex is in this approach, this focal graph approach, is essentially an agent. It's a search agent that can search biomedical data in a unique way, as we see it. And so when you combine that with large language models, now you can we've already shown that you could take a large language model and feed it a prompt and say, here's how focal graphs work or Plex works. Here's the strategies you might want to use. And a large language model can actually plan a research strategy using this sort of approach, right? So it can plan the strategy. We also have shown that given the prompt with the background, it can interpret the results as well. So you could run your search and you find the stuff and it can look at it and say, here's what's promising in this result for curing cancer. Here's the key summary. And then in between that, it's pretty trivial to have it also just run the search. So now you have all the pieces. It can plan a research program. It can run searches. It can interpret searches. Could even adjust its plan based on the result and run another search and another search. And this can be scaled. So we've already covered that this kind of system can find novel discoveries, can make novel discoveries transparently. And the planning also of the large language model is transparent. So if you put this all together, and this is what I think is really mind-blowing about this whole picture, you put that all together, you have a largely autonomous, or at least partially, but a largely autonomous scientist that can plan research programs, can execute them, can do this at scale, and can deliver novel findings that no one knows, and the experimental data that supports them. So again, not a suggestion based on papers it's read, but actually findings based on the data that we've collected, and provide the actual data and all the details and the methods it used, right? What the research program was, the strategy it used, all of these things. So it really, I think it foreshadows a potential truly autonomous AI scientist, which I think is incredibly exciting. People are talking about this, debating whether that's possible and when and how that might look. Is the best that I can tell, we've assembled all these pieces. In fact, in our paper, we show it working at least a little bit. We show it running for 10 minutes or so, right? And finding new things, finding something new, actually a few new things in the cancer space. So I find that tremendously exciting. I think that's really going to have a massive impact on this field. It's going to change the way we approach research. Now scientists are going to be utilizing this kind of tool, this kind of approach, supervising, guiding it, optimizing it, refining it, making sure that it's finding things that are useful, that make sense, that pass regulatory muster, right? Which is where the transparency is really critical. And I think it's going to really enable a revolution in this space. And so that's the big message we're trying to get out there, is to get people talking about it, because I think people haven't put all the pieces together yet. And I think all the pieces are there to put together. And that's trying to tell that story.
Are you referring specifically to the paper that was published by a research team at Google? Because I think they call it an AI scientist.
Yeah, they called it, like Google called it the AI co-scientist.
Yeah, AI co-scientist. Yeah, I've seen that. And I would agree that's a very exciting thing. And I think the other thing that I find quite exciting, and you talked a bit about it, it's the bridge between the virtual and the physical world. So there is all the planning, and then you connect that to automation, to lab automation, and you execute it. And then eventually you get the data back into the system in a semi or fully autonomous way. And that, that sounds like science fiction, but actually it's working now, which is mind-blowing, I think.
Yes. Oh, it's incredible. That certainly can be integrated so that now you've taken it as far as you can with existing data, which could be really far. I think that's going to really surprise people, how far we can go with the data we already have. But there's always more to do, right? So now you get to the very end of what we can do with the data we have. So now the system says, okay, what data will get us the furthest, that will continue the success, continue the discovery, tell us what we still don't know. And it will feed back and say, this is the kind of experiment we need to run now. And potentially even that could be automated. It could be the kind of experiment that could be automated. So that gets run, and now the data that will push us further ahead gets generated automatically.
To close this conversation, obviously this field is developing super fast. Yeah. I spend hours every day trying to stay up to date, and obviously even if I do that, I'm missing tons of information every day. Are there anything that you would recommend to people who want to stay up to date on what's going on, things to read, people to follow, any other suggestions?
That's a great question. I don't know. I'm having trouble staying on top of it. I think everybody is. That's really good at that saying. Maybe the filter is if you're a scientist, you can't drop your principles, right? So you need to know, there's certain things that science requires. And so you keep that in mind, and that becomes a filter. So there could be a lot of things out there that are just, so lack of transparency and these kinds of things. And there's this current skepticism. I think a lot of scientists are pushing it away until they see what they need, until they see the right level of transparency and things like that. And I think it's okay to use that kind of thing as a filter, especially if you're going to be the one consuming it in the end. Obviously, if you're actively working in the field, then you need to be aware of that gap, where there's a limitation of your technology and the need, right? So you can work on it and refine it and try to turn it into something that's useful. But I think if you want to be an informed consumer of AI, just be clear about what it is required, what the technology is actually delivering, what that gap is. Be clear about it and keep an eye on places where the gap is closing, right? Approaches that are getting closer to what is required. And maybe on the other side also, some of your requirements, be careful that are they real requirements, right? Do they really need all of these things? Or perhaps there's some things that this technology is delivering that really are useful. I just need to change my thinking a bit, right? So it's probably going to require flexibility on both sides, of the technologists as well as the consumers of the technology. There's definitely a little bit of meeting in the middle, but I don't know the answer to how do you avoid the overload. For a lot of people, it's you throw it all into an LM and have it summarize it for you. So I'm sure that's part of it. And I certainly use that as much as I possibly can. But yeah, there's a tremendous amount of stuff coming at us right now, which is very exciting, but also pretty overwhelming.
So last but not least, if people want to know more about Plex and the latest news, do they follow you on LinkedIn? Where do they go? Yeah,
LinkedIn, certainly. Plex Research is our company. plexresearch.com is our website. If you want to dig into it, so the preprint that we have, and we have this prominently on our website, so that AI framework paper, and we'll link it as soon below. So that's a great place to take a look. And I would encourage you, if you do go there, the supplemental files are worth looking at. One of them is a transcript of asking our system with the large language model, hey, can you find a new way to cure cancer for me? And it's a certain type of cancer. We're a little bit specific, but it's a really very pie in the sky request. And the system goes off and does and tells you what it's doing and tells you precisely what it found, and it makes demonstrable progress. Even if it's only the first step, it makes demonstrable progress and verifiable progress towards that. So I think that's very exciting. And then even the other supplement is comparing if you have a large language model without this sort of approach versus with it, the difference is just really pretty amazing. So to have a look at that, and just, I'm curious about beyond biologists and chemists and people in this space, I hope it's accessible to a wider audience. It was intended to be that way. And if you're interested in having a look and seeing if it resonates with you, if some of these ideas really catch your eye, catch your interest, please have a look and let me know.
Yeah, thanks. I think that was incredibly exciting, incredible development, and certainly more to come. So thanks a lot for the discussion.
Fantastic. I enjoyed it very much.